Module 4, Objective 7, summarize the excitation-contraction coupling and the contraction cycle. The contraction cycle consists of 6 discrete steps. Here they are. It begins with the arrival of calcium. Remember, calcium has been released from the sarcoplasmic reticulum due to the arrival of the action potential at the triad. As calcium is released, it diffuses through the sarcoplasm, and it's going to arrive within the zone of overlap of the sarcomere. Step 2, active site exposure. Calcium binds troponin, and that causes the troponin / tropomyosin complex to change position, change shape. In doing so, it's going to expose, let me get my ink pen, it's going to expose these active sites on actin. These active sites are also called the myosin binding sites. So as you can see back here in step 1, these active sites are hidden. They're being blocked by tropomyosin. But as troponin is bound, as calcium binds troponin, the troponin-tropomyosin complex shift, they move, and now those active sites are exposed. And that's going to lead to cross-bridge formation, which is step 3. Cross-bridge formation occurs when myosin binds actin, when these two molecules bind. Okay, so that's cross-bridge formation. Step 4, the pivoting of the myosin heads. Once you have cross-bridge formation, the energy that was stored in the resting state of the myosin molecule is released, and the myosin heads are going to pivot. They're going to change shape. They're going to move in this direction. In doing so, you get the formation of the power stroke, okay, the pivoting or moving of that myosin head. And the power stroke, when that occurs, the bound ADP plus P, right? So ATP is transformed to ADP plus inorganic phosphate. That's going to be released, and we can see that happening right here. Another ATP molecule is going to bind myosin heads. It's going to be hydrolyzed, so it's pretty much the reverse reaction up here. It's going to be hydrolyzed. That is going to recock the myosin head. It's going to re-energize it, take it back to its cocked position. At that point, you get a breakage between the two molecules. All right, so let's sort of formalize this. We'll start with step 5. When another ATP binds the myosin head, the link between the myosin head and the active site on the actin molecule is broken. So the two molecules separate. The active site is now exposed and able to form another cross bridge. Step 6, myosin head reactivation occurs when the free myosin head splits ATP into ADP and an organic phosphate. The energy released is used to recock the myosin head. When muscle cells contract, they produce tension or pull. In order to produce movement, tension must overcome the resistance or the load or the weight. The entire muscle shortens at the same rate, mainly because all the sarcomeres contract at once. The speed of shortening depends on cycling rate, which is the number of power strokes per second. There are 3 things that really determine the duration of a contraction. All right, so if you were to pick up a dumbbell and flex at the elbow, so your biceps brachii are going to flex, and you're holding that dumbbell, 3 things determine how long or the duration of that contraction. One is the neural stimulus. You could just say the heck with it and drop it. So that would be one thing that determines the duration of a contraction. Second, the availability of ATP. As long as you have ATP, you can energize those myosin heads and maintain a state of contraction. But as ATP runs out, contraction stops. If you've lift that dumbbell and you lift it until physically you can no longer lift it anymore and it falls, most likely it's the lack of availability of ATP. You used up all of your stored ATP and there's just none left. The other thing that could affect the duration of a contraction is the presence of free calcium ions within the cytosol. You have to have the free calcium there in order to bind to troponin, in order to expose those binding sites, those myosin binding sites on actin. So 3 factors that contribute to the duration of a contraction. Relaxation, as calcium is pumped back into the SR. Remember, calcium is stored in the SR and it's actively pumped back into the SR for storage. The calcium concentrations within the cytosol falls. As those calcium concentration falls, calcium detaches from troponin, tropomyosin, and troponin return to their original position, covering the active sites on actin. So at that point, the contraction ends. Rigor mortis is a fixed muscular contraction that occurs after death. It results when ATP runs out and the ion pumps stop functioning. So here we're talking about calcium ion pumps on the sarcoplasmic reticulum. If you don't have ATP, you can't pump the calcium back into the SR. So calcium concentrations build up in the cytosol, causing muscle contraction. And it occurs hours after death, and it can last for hours. But the term stiff to describe a dead person came from this process, this physiological process of rigor mortis, fixed muscular contraction after death. So the contraction cycle as a whole, if we were to try to summarize this, let's do 2 things. Let's look at the image first and kind of walk through, and then we'll look at the verbiage and allow it to match it, or maybe we'll do it at the same time. So here's your axon terminal, okay, the end of the neuron. So if we were to sort of draw that out, it would look like that. So the axon terminal. And we know that as the action potential travels down the axon, it's going to cause the release of acetylcholine from the axon terminal. Acetylcholine is going to diffuse across the synaptic cleft. It's going to bind its receptor on the motor end plate. These receptors are sodium ion channels. And as acetylcholine binds sodium, these channels open, sodium floods in, and it leads to the generation of another action potential. And we can see that here, traveling along the sarcolemma, down the T-tubule, causing the release of calcium at the triad. Calcium concentrations in the cytosol increase. They're going to diffuse. They're going to bind troponin. They're going to cause the tropomyosin molecule to shift out of the way, allowing myosin to bind actin, forming a cross bridge. With cross bridge formation accomplished, the myosin is going to undergo power stroke, and it's going to systematically pull the thin filaments across the thick filaments. It's almost like a tug of war. All these little myosin heads are going to grab onto these thin filaments, and it's going to pull in this direction. And as you get pulling from both sides, the Z lines, right here and right here, they become closer together, and you get a shortening of the sarcomere. And as the sarcomeres shorten, the muscle shortens, and it's going to create tension at the tendons, which are pulling on bones. There are a lot of YouTube videos, if you were to search neuromuscular junction, if you were to search any of the main topics that we covered here, excitation, contraction, coupling, you're going to find a lot of nice videos that are animated that may do better justice to this topic than my piddly drawings. So I would highly recommend spending some time watching those to help get all this information down.